Okay, let's continue on. Chapter 9. I skipped over the part in the book about the genetic basis of short legs in dogs. We don't have to go there. And talking about genetic testing like amniocentesis, I've already mentioned this before about the probability of having a non-disjunction event and therefore having a Down syndrome child is much greater for women who are pregnant, they're over 35. So I mentioned before her obstetrician will almost certainly suggest that you get amniocentesis, where you withdraw cells from sucked off cells from the fetus, squash them, and look at the chromosomes and see and count how many chromosomes there are. Okay, get back to the Mendel's. Now we're talking about Mendelian traits, right? They explain inheritance in terms of two genes that pass along generation to generation according to simple rules of probability. And these laws are valid for all sexually reproducing organisms. But Mendel's laws don't explain everything. Things are a little bit more complex, actually a lot more complex. For most sexually reproducing organisms, cases in which Mendel's rules are strictly account for the patterns of inheritance are actually kind of rare. More often, the observed inheritance patterns are more complex. So now we're going to look at what are called non-Mendelian traits that Mendel didn't discover. Okay. One is called incomplete dominance. Okay. In Mendel, a Mendelian locus has complete dominance. One allele is completely dominant over the other. In incomplete dominance, and so you only have two phenotypes: the dominant expression trait and the recessive expression trait. That's it. But in incomplete dominance, the F1 hybrids have a appearance that's exactly halfway between the two parents. It's also known as blending inheritance, where the heterozygote has a different phenotype than the homozygous dominant. Remember, in complete dominance, big B, homozygous dominant and heterozygote have the same phenotype. In incomplete dominance, and so there's only two, and remember, there's only two phenotypes, but in incomplete dominance, there are three phenotypes because the heterozygote has a different phenotype than either the homozygous recessive or the homozygous dominant. And we call it homozygous dominant, even though, so neither allele is completely dominant. Okay, hypercholesterolemia is a human trait, the example of incomplete dominance. It's characterized by dangerously high level of cholesterol in the blood. Heterozygotes have blood cholesterol about twice normal. Homozygotes have about 5 times the normal amount of blood cholesterol, and may have heart attacks as early as age 2. If you're a heterozygote, you're going to have a heart attack probably in your 40s, maybe your 50s. A homozygote, you're going to be a little child going to have it. So this is what the statin drugs have been developed for. And here is incomplete dominance in snapdragons. It's even easier to see in snapdragons. So there's, unfortunately, they give you big R and small r to talk about two alleles. And in this case, big R and small R doesn't mean big R is completely dominant, small r is completely recessive. So you have the parental generation, red, big R, big R. The white is small r, small r. They got to make the heterozygote pink, exactly halfway between the two parents. What happens if you mix red paint and white paint together, you get pink. And then you make two pinks together. And now you have, and with the F2 generation, you have 1/4 red, big R, 1/4 white, small r, and half pink, the heterozygote. So, like I said, as soon as you see that the heterozygote has a different phenotype, automatically, you know it's incomplete dominance. Right? And there's incomplete dominance of human So LDL is the bad cholesterol. The level of LDL in your blood is a direct risk for having a heart attack. And so there are LDL receptors on cells that bring LDL into the cells and take it out of the blood. So it lowers the amount that's in the blood. If you're big H, homozygous for ability to make LDL receptors, and your cholesterol is normal. Small h, homozygous for inability to make LDL receptors. You don't have any LDL receptors. Severe disease, heart attack when you're toddler. But if you big H, small h, heterozygous, you have half the number of LDL receptors as normal. Therefore, your LDL cholesterol is twice what is normal, and you're at high risk for having a heart attack in middle age. So there's the genotype, and here's the phenotype. So like I said, you can't always tell by looking at a person what their phenotype is, because it could be microscopic or in cells, and you can't really tell them. Like I said, like blood type, you can't. My phenotype is A positive. You can't look at me and tell me I'm A. You tell that I'm A positive, but it's still a phenotype. And speaking about blood type, there we go. Now, the ABO blood type in humans involves 3 alleles of a single gene. Okay, this is called multiple alleles. If you, in Mendelian loci, only 2 alleles are possible, right? Big A and small a, for example. But in multiple alleles, more than 2 are available. In the ABO blood typing in humans, there's 3 possible alleles: A, B, or O. And they produce 4 phenotypes: blood type A, B, AB, or O. Now, I said I'm A positive. Well, now, the positive or negative is called the rhesus factor, and that's a different locus than this locus. And it is a Mendelian locus. There's only 2 alleles possible that produce the Rh factor, which is a protein on your red blood cell. It says it's carbohydrates. It's a glycoprotein. It's that are on the surface of your red blood cells. That's what the A is. The A protein, or glycoprotein, the A glycoprotein, the B glycoprotein, and the no glycoprotein is O. So if your blood type A, you have the A glycoprotein, and that's it. If your blood type B, you have the B glycoprotein, and that's it. If you have, if your blood type AB, you have both of them. And here's another characteristic that this locus what's the word I'm looking for? Exhibits. It's called codominance. Sometimes you have more than one allele is dominant. And in an ABO locus, A is dominant and B is dominant. So if you get an A from one parent and a B from one parent, your blood type AB, and they're both present. O is the only one that's negative. So O is the absence of those glycoproteins. If you type O, you have neither glycoprotein. But that is recessive. You have to get an O allele from both parents in order to have O, and it's recessive. Sometimes it's, usually when I use it in class, I would designate the O allele as small I. So you have to have 2 small i's to have blood type O. Okay. Because there are 3 alleles, there are 6 possible genotypes. Okay, and yeah, they use small I here also to designate the O allele. So you can have a genotype of A, I, and your blood type A. So there's big A is blood type A, big A, I is blood type A. Blood type B, big B, or big B, I. Right? With blood type AB, there's only one genotype possible, AB. So it's the same as the blood type. That's your genotype, AB. And then for O, there's only one possible genotype, and that's II, two small i's. So, and here to talk about codominance also. So if you make both glycoproteins, your type AB, you have both of them, and they're both dominant, so they both are expressed. Okay, here's the phenotype AB or O. Here's the genotypes. Two possible genotypes for A, two possible for B, one for AB and one for O. And here's the strange thing. Okay, antibodies present in blood. Here's where it comes down to blood transfusions. This is really bizarre, and I don't know why this is. We've talked a little bit about immune function and making antibodies. And you probably know about antibodies because of being vaccinated for COVID-19. That means we can make antibodies, these immune proteins, against COVID-19. So I just got my second Pfizer vaccine like a few weeks ago, so I'm feeling pretty good. Because now I have these antibodies in my blood against that coronavirus. They're there. But here's the thing, if I hadn't have been vaccinated, I wouldn't have those antibodies. Now, a person who gets coronavirus and and lives through it, they have the antibodies now, right? Just like they've been vaccinated. That's called artificial immunity versus if you get the disease and you live through it and you have antibodies present, that's called natural immunity. If you get vaccinated for it, you also have the antibodies for it, but you got it artificially, so that's called artificial immunity. Either one protects you, right? But the important thing is that if you don't come into contact with the coronavirus or you don't get vaccinated for coronavirus, you don't have anti-coronavirus antibodies in your blood. But if I'm blood type A, I just said I was blood type A, I have anti-B antibodies in my blood. I don't know why. They're pre-made. Even though I've never been exposed to the B glycoprotein, I have antibodies against them, like pre-made. So here's why I can't get B blood. Here, if I need a transfusion, they're going to give me A, right? If they give me B, what will happen will be agglutination will happen because the antibodies are Y-shaped and they have two binding sites for B. So they can bind a red blood cell with B, another red blood cell with B, another red blood cell of B over here, and it forms big clumps of red blood cells clumped together, and it blocks arteries and veins all over your body, and it causes death. That's death from mismatched blood. So I cannot get B blood; it will kill me. I can't get AB blood either; that would kill me because B is present. However, O blood, B isn't present, right? So when I pinch, I can get O. That's why it's called O, the universal donor, because that person has neither A or B. So in a pinch, I could get it. Now, if I need a transfusion in the hospital and they got A blood, they're going to give me A. But if they're completely out of A and it's an emergency situation, they can give me O. They can't give me B. So if you're B blood, you have anti-A in your blood, so you can't get A blood, but you can get O also. If you're blood type AB, that's called the universal recipient. He has both A and B, therefore he has neither antibody in his blood. So he can get anybody's blood safely. But I has both antibodies in his blood, anti-A and anti-B. So O can only get O. Can't get A, can't get B, can't get AB. It'll cause agglutination and death. So so O is the universal donor, can give to everybody, but can only get O. AB is universal recipient. He can get blood from anybody and it will be safe. Now remember, the Rh factor is a Mendelian locus, and it acts just like coronavirus. If you're A negative, for example, you don't have anti Rh factor in your blood. It's not there. So if you're A minus, you could actually get A positive for the first time only, and it'll keep you alive. But now, since you've been exposed to the positive, now you have anti-Rh in your body, and you can't get another injection of A blood. And this is a problem for women, pregnant women who are negative, and they have a positive child, because there is a little bit of mixing of blood at birth, and so that this woman who has a negative blood type comes in contact with the positive Rh factor in your infant's blood, and she makes anti-Rh antibodies. And so if she has a second child that's also positive, those antibodies will cross the placenta and kill the fetus. So that's why women who go into the hospital to give birth and have a negative blood type are given this injection called Rogan, that's anti-Rh antibodies, pre-made, to destroy any kind of Rh positive blood that gets mixed with hers before it goes to her lymph nodes and she makes anti-Rh antibodies. Okay. So there's the same thing right there. Those are the reactions. These are agglutinations here and would cause death. And you can do, have your blood tested easily to find out what blood type you are if you don't know. Okay, as I just said, matching compatible blood groups is critical for safe blood transfusions. If the donor cells have the glycoprotein A or B, it's forwarded to the recipient's immune system produces blood proteins called antibodies. Well, no, they're already made. They're already there. That's the really bizarre thing. And these antibodies cause the donor's blood cells to come together. So Maria has type O blood, and her sister has type AB blood. What are the genotypes of the girl's parents? All right, actually, let's do it on the board. So, Maria, let me stop sharing for a moment. As O, and what was the other sister? As AB. So what do mom and dad have? Well, remember, this can only be II. So mom and dad have to both have a small I. Okay, sisters AB. So I got an A from one parent and a B from another parent. So one of these parents is blood type A and one is B. You don't know which one. They have to be that in order to get one sister with this and one sister. Can you see that? So I like to do have fun with paternity tests. Okay. Mom and dad. Because paternity tests, women always know who their children are. It's kind of obvious, right? But dads, they can't be absolutely sure, right? So mom is blood type A. Well, this was the same situation. Dad is blood type B, right? They have a child, say Tom was the child, whose blood type is II. Um, so should dad worry? Could did mom fool around? Can he have an eye child? Well, yeah, if he's like this, as we just saw. But if we test him, it turns out to be this, mom's fooling around. There's no way. Or if dad were AB, and they have and the child is O, can that be? No, mom's fooling around because he doesn't have an eye to give, so he can't have an O child. Okay, let's go back to the screen share. Go back to the slideshow. Oh, wait a minute, where are we? We're on here. Okay. Oops. So let's go on to some more non-Mendelian traits. Pleiotropy. So one gene influences several characters. So remember, Mendelian is one gene, one character. One gene, one character. Pleiotropy is when one gene affects more than one character. A good example is sickle cell disease exhibits pleiotropy. Results in abnormal hemoglobin protein and causes disc-shaped red blood cells to deform into a sickle shape with jagged edges. It's much more common among Africans and African Americans than among Caucasians, for a reason I'll talk about in a moment. So it causes abnormal hemoglobin proteins. It also exhibits incomplete dominance, and that is if you have two small letters, you have severe sickle cell disease, and you're probably going to die as a very young child. If you're heterozygous, you have sickle cell trait, and it could be debilitating, and it's tough, and it could be painful, and you got to be careful about too much exercising or going too high of an altitude, because if you get into a low oxygen situation, your cells become this sickle cell shape, and it blocks blood vessels, and it's really painful. But you can live. So if you get into a low oxygen situation, the cells don't flow smoothly in the blood, and it tends to accumulate and clog tiny blood vessels. So altering structure affects function. And there it is. So sickle cell neuron. So under a low oxygen situation, they become sickle shaped rather than dumbbell shaped. But they don't, for some reason, they don't mention the other trait. This is kind of a flaw in the book here. One trait is causing cells that become sickle shaped in low oxygen situations. What's the other trait? The other trait is protection against malaria. You cannot get malaria if you have sickle cell trait. So it protects you. And that's why it's so more common among people of recent African heritage. As remember, all human beings have African heritage, because that's where human beings came from. As we've talked about before, about the evolution of skin color. But recent African ancestry, more likely to have sickle cell, because in West Africa, malaria is all over the place. It's abundant. The mosquitoes that carried are everywhere. So everybody gets bitten by these mosquitoes. And so so here's the thing. If you're two small letters and you have severe sickle cell disease, you die from sickle cell disease when you're a little child. If you are two big letters and completely normal hemoglobin, you're going to die of malaria. So the only, and this is evolution at work here. And so the only people that survive are the heterozygotes that have sickle cell trait. Now, so they have to be careful about being in low oxygen situations, but they can survive and live relatively normal lives. And so they are selected for in evolution, and that's why it's much more common in people of recent African ancestry. So which major theme is illustrated by its actions, relationship of structure to function, as you'd already said before. All right, polygenic inheritance. Exactly the opposite of pleiotropy. Additive effect of two or more genes in a single phenotype. So here, one trait controlled by more than one gene. So pleiotropy was one gene controlling more than one trait. Polygenic inheritance is one trait controlled by more than one gene, usually many genes. So how can you detect whether something's pleiotropy? Well, if you have so many phenotypes in this trait that it fits a normal distribution, a huge number of phenotypes, then that's probably polygenic inheritance. So human height. How many different phenotypes are there? I mean, a gazillion, right? Depending on how small you want to make the differences, you know? So this is polygenic inheritance. Skin color also, we just talked about skin color. That's also polygenic. How many different shades of skin color do human beings have? Lots and lots. So they exhibit polygenic inheritance. And some diseases, diabetes, heart disease, cancer, polygenic inheritance. So it's an example of the theme of interactions, how that a new property can result from the interactions of many smaller parts, genes. So there's the normal distribution right there for height. So here's the fraction population. Here's the height from very short. Now, how short can you be and still a normal height? Because we talked about before dwarfism. But the shortest people that are normal height probably around like 4'10", 4'11", something like this, I think. To very tall. How tall can you be would be a normal tall and not be gigantism. And that's probably like, I don't know, 7'6", 7'7", minute bowl type height. Above that, and that's like gigantism. But there's just so many because like it could be 8 genes controlling height, and all these different combinations lead to all a different phenotype. So that's polygenic inheritance. Well, no, this is only 3 loci. And that's how many different variations you can have. I think it's actually more than 3, but anyway. So I'm never going to give you a Punnett square that has 3 or more genes that control it. There's the normal, that's called a normal distribution. So we got an average height, and then very short and very tall, and lots of phenotypes. Okay. So your skin color is driven by many different proteins acting together in combination with environmental factors. Well, you have a certain genetically set skin color, and depending on how much time you spend out in the sun, it's going to make it darker or lighter, right? So which major theme is illustrated by this interactions within biological systems? Oh, okay. So many phenotypic factors result from a combination of heredity and environment, like we were saying. Sometimes the environment will affect the expression of genes. So this is the nature and nurture controversy. So spending time with identical twins will convince anyone that environment, which can include effects of personal choices, affects a person's trait. In general, only genetic influences are inherited, and any effects of the environment are not usually passed on to the next generation. For example, your height, we're just talking about height, is genetically determined, right? You're going to be a certain height. But what if you have poor nutrition as a child? Will you reach that genetically set height? No, you'll be shorter. So here how the environment can affect that. This is called epigenetics, EPI genetics, epigenetics. Wait a minute, there's another term also, which I forgot. I'll bring it up in the next video that is also a word that means the effect of environment on the expression of genes. So as a result of environmental influences, including choices such as hairstyle and color, even identical twins can look different. If you produce clone mice, how would you predict the number of epigenetic differences among the clones to change as they age? Well, you can look at the number of different things they're doing to look at the epigenetic changes. Okay. Okay, so here's epigenetic inheritance. This is how our biologists become to recognize the importance of epigenetic inheritance, the transmission of traits by mechanisms not directly involving the DNA sequence. Components of chromosomes can be chemically modified by adding or removing chemical groups on DNA or protein components on chromosomes. Epigenetic modifications and the changes in gene activity result may even be carried on to the next generation. Some of these epigenetic changes can actually be carried on. The mechanism of that I'm not sure how it is, because maybe it's because of these chemical changes to chromosomes. These chromosomes are passed on to your offspring and they've been modified by you, so this epigenetic change is passed on. So the chromosome theory of inheritance states that genes are located at specific positions, loci on our chromosomes, and the behavior of chromosomes during meiosis and fertilization accounts for inheritance patterns. These chromosomes undergo segregation and independent assortment through meiosis. So figure 9.24 correlates the results of the dihybrid cross in figure 9.8b with the movement of chromosomes through meiosis. Okay, and this is, like I said, this looks really complicated. This is what we were talking about before. I think I already explained this. So this is the R locus and the Y locus, and how they arrange in metaphase 1 determines what side the big R's are and what side the small R's are, and what side the big Y is on and what side the small Y is on, just by the way they arrange in metaphase 1 of meiosis. And like I said, if they're on different chromosomes, they're going to independently assort, because each pair of homologous chromosomes assorts independent of any other pair of homologous chromosomes. So we already really went over this. And we get the 9:3:1 ratio. We already did that with round versus wrinkled and yellow versus green. For each pair of homologous chromosomes, one member derives from your mother and the other from your father, ensuring the transmission of genes from one generation to the next. And this is evolution, I think, because evolution is a transmission of how each generation changes because of the transmission of genes from one generation to the next. Okay, linked genes, I already mentioned this, located near each other in the same chromosome and tend to travel together during meiosis and fertilization. So linked genes are often inherited as a set and do not follow Mendel's law of independent assortment. So far, the patterns of genetic inheritance we've discussed have always involved genes located on autosomes, not the sex chromosomes. Now we're ready to look at the role of sex chromosomes and inheritance patterns exhibited by characters they control. In other words, the next big topic, sex linkage. And that we're going to hold off on that until the next video lecture. So this is 106 genetics 3. And we're going to need at least one more to finish this chapter. So I'll catch you on the next video.